Hello to our friends joining us via recording. Today we are starting our discussion of lesson number 6. Lesson number 6 has a really weird title. So if you look at that, it's called The Central Dogma. And I don't know about you all, but when I was in college and I first saw this terminology, the central dogma, I was like, oh my gosh, this must be like the craziest thing ever. It sounds super confusing and scary. I think it's because of this word dogma at the end. I had no idea what it meant. But the central dogma actually isn't as scary as its name sounds. As we're going to talk about today, the central dogma is the way that your cell takes its genetic information that we store in the form of DNA, and we turn that genetic information into proteins. If you remember back from unit number 1, we said that cells have a lot of proteins inside them. We actually said there were a lot of different reasons that your cells had proteins inside. Can you help me out in the chat for my friends who are here? What were some of those things that we said that proteins did for a cell? Why does a cell need to have so many proteins? Yeah, so the first one that we see a lot with neurons is that they help our cells to have their shapes, right? So if you remember, neurons are cells that look really funny. We saw them in lab when we were looking at the slides. They look kind of like stars. It's because they have so many proteins inside of them that they have that odd shape. Or we use proteins to act as enzymes. Enzymes are things that make a reaction happen more quickly. When we talk about this process of turning DNA into mRNA, there's a special enzyme that does that. Well, actually, there's multiple that help us to do that. So we use proteins to get things done, to work as enzymes. Yeah, I also see a comment in the chat about allowing us to transport things across the plasma membrane of the cell. Here, I'm going to make us do a little bit of review to help us never forget. When we talk about moving something across the plasma membrane of a cell using a protein, would I use the chemistry word polar or non-polar to describe something that has to go through a protein to get across the membrane? Polar or non-polar? Yeah, polar things, I see several of us in the chat there. Polar things are the kinds of things that don't match the middle of the membrane. If you don't match the middle, you need a protein to get through. So polar things use some of these proteins that we're building to get across the membrane. Yeah, we also had a comment about cells using proteins to communicate with each other. Absolutely, because it's on the outside of their membrane, so they can talk to their neighbors. Proteins also do stuff. The one other function that was mentioned in the lesson is that they help a cell to do its overall job. The example we gave of that was hemoglobin. And I mentioned that again because we're going to talk about how it's really, really important that the central dogma be correct when we're building proteins. Because if we have an error in this process of building our proteins, whether it's a problem with the DNA or a problem with the RNA, we can end up with a protein that doesn't do the right job. And that's what happens in sickle cell anemia, is we have an issue with how the central dogma is working. So hopefully by the end of today, we'll get to the point where we can talk about that. But the other job of proteins in the cell is to do big picture things, like transport oxygen. We've got to build the protein correctly for it to be able to do its job. So lots of important reasons that we need the central dogma to work. And again, I promise it's not as scary as its big long name seems to make it sound. To help us get ready to talk about how we build proteins in the cell, we started off with our groups to do a little bit of review about proteins. So let's work together as a big group now. We're going to work through these questions one by one to remind us of some of the most important stuff from lesson number 5. One of the first things we talked about in lesson number 5 was this word monomers, or the small pieces that I use to build a protein. Who can remember the name of the monomers, or those small pieces that we use to build proteins? Yeah, exactly. When we talk about the monomers, or the little pieces of proteins, we're talking about things called amino acids. Amino acids. Here, I'm going to do a terrible job here, but we're going to try to draw. So here's my amino acid, right? I've got a couple of different extensions. So on one side of my amino acid, I'm going to have a carboxylic acid group. On one side, I'm going to have an amine group. That's why it's called an amino acid. And at the other part of it is what we call a functional group, or our R group. Remember, was the other thing we called it? We didn't actually use this terminology during class yesterday, but what did I tell you to kind of think about those R groups? Like, do you remember? What kind of thing does an R group tell us about an amino acid? We may not, I'm doing a bad job of asking this question. So yeah, there we go. At least one person picked up on what I was saying. When we talked about the R groups, or the functional groups of the amino acids, remember that I told you to kind of think about them like the personality types of our amino acids. So we have the amino acids that are polar. Their personality type is they're polar. We have the amino acids that their personality type is nonpolar, or that their personality type is negative or positive. So the personality type of an amino acid, that's this last part here, my R group, or my functional group. And then we've got the acid and the amine groups. All of those parts of our monomers, or our small pieces, called amino acids. To build a protein, to connect my amino acids together, I have a special kind of chemical bond. And I told you, anytime you hear the name of this bond, we should always think protein. Do we remember the name of that special kind of bond that's only in proteins? Yeah, several of us chiming in here. The kind of bond that's only in proteins, those are called peptide bonds. Peptide bonds. Remember that a peptide bond forms when we steal water by taking a hydrogen from the amine group and an OH from the acid group. We do what's called dehydration synthesis. Dehydration synthesis. That's the process that I use to build a peptide bond. I dehydrate, or steal water from my amino acids to attach them together in a peptide bond. So if we look over at our picture over here, we start off with some amino acids that are floating around, they're not connected. Then I add some peptide bonds to connect them together. Our question asked for the name of the level of structure in a protein that I see when I look at the order that my amino acids are connected to each other in. When we talk about a line of amino acids connected with peptide bonds, what level of structure do we call that? Yeah, exactly. That's what I call my primary structure. Primary structure. So remember, the primary structure of a protein, it's the order the amino acids are in. The order that the amino acids are in. Hey, remind me from our class yesterday, when we talk about the primary structure of a protein, the order that its amino acids are in, is that going to have any effect on my other levels of structure? So does primary structure, is that important or not important for figuring out the other levels of structure? Yeah, for sure. All of us are chiming in and saying, absolutely, that is important. Remember that if I mess up my primary structure, if I somehow make this wrong, if my order of amino acids is wrong, I'm going to have issues doing any of my other kinds of structure, or I potentially will have issues. I might still get the same protein, I might be okay, but there's a very good chance I will not be okay. And that's what we're going to talk about with hemoglobin at the end of class today. I heard some good conversations with my groups about which level of structure I see right here. Let's suppose that what I'm looking at right here is what we call an alpha helix. If this is showing me an alpha helix, which level of protein structure would I call this? If C represents what we call an alpha helix, let's say that that's what it is, for example. Yeah, if this is showing me that curly Q ribbon form of a protein, part of a protein, we would call that secondary structure. Secondary structure, if we're looking at an alpha helix. It's possible, though, that we're not just looking at an alpha helix. Maybe we are looking at the overall 3D shape of this protein. Yeah, and that's what Kaylin's mentioning. Perhaps this is not just an alpha helix. Maybe this is the whole 3D shape of our protein. If this is the overall 3D shape of my protein, what would we call this one if it's the overall 3D shape? Overall 3D shape. Yeah, if we're talking big picture and it's all the 3D stuff, now we're doing what's called tertiary structure. Absolutely. Tertiary structure. I would accept either of these answers. So either of these would be correct. Yeah, Gerard was saying we got some weird perspectives going on there. Absolutely. Which is why we can make a case for either of these levels of structure. So just remember that if we're talking about just part of an amino acid chain and it's in an alpha helix, or the other one was called a beta sheet, either of those were examples of secondary structure. If we're talking about this is the entire protein right here, and this is the way it folds overall 3D, that's going to be tertiary structure. Tertiary structure. Hey, on that vein, when we're talking about the 3D shape of a protein, the last question here asks, there's a name for a protein that has permanently lost that 3D shape. What's the name for a protein that has permanently lost its 3D shape? Yeah, exactly. Denatured. Denatured. Remember from yesterday, I told you all this is an underline, highlight, star word. We must know this word denatured. It means that my protein has permanently lost its shape. And remind me, if I have permanently lost my shape, can I still do my job? If I permanently lose my shape? Yeah, we're right. We're saying in the chat, if I permanently lose my shape, I permanently lose my function as well. So yeah, Tien's reminding us that the everyday example of a denatured protein that we've seen in our life before is when you fry an egg. So it goes from being clear to being white, the whites of the egg. Those proteins are permanently changed. We're not going to be able to get them clear again. And we're also not going to be able to have them do their function for us anymore. So no more growing baby chickens in that albumin. So if we are denatured, we not only have the wrong shape, we also have the wrong function. So that's a good overview for us of a lot of the material from lesson number 5. Before I move on, any questions about this activity that we did together? Or shoot me a thumbs up if you feel good about it. Perfect. Okay, so I'm getting a whole lot of thumbs up. That's great. Let me take a moment to remind you that the homework assignments, there were 2 of them for lesson number 5. The one that relates just to the stuff we talked about in class, and the one that relates to Creutzfeldt-Jakob's disease, so the protein folding and disease assignment. Both of those assignments are due on Sunday. So make sure that we're wrapping up lesson number 5 by Sunday at the latest. We got some more time on lesson number 6. That one is not also due on Sunday. That one's been bumped a little bit, but we're going to start talking about it today to make sure we have plenty of time to digest it and work our way through it. All right, so let's talk about lesson number 6. Remember that we mentioned at the very beginning of our class time that what we are talking about here in lesson number 6 is called the central dogma. The central dogma. So the central dogma, this is kind of like the one thing that cells have to be able to do to stay alive. They have to be able to use their DNA to build polypeptides. Now, this is a slightly different word than what we talked about yesterday. Yesterday, the word that we used that's similar to this is we called proteins a polymer. This word poly just always means there's more than one. Can we take a guess here? What do you think this peptide part is referring to in this word polypeptide? I promise it's not a trick question. Polypeptide. Yeah, so a couple of us are honing in on a word from the last slide. We talked about peptide bonds. A polypeptide is a molecule that has more than one peptide bond. And remember that peptide bonds are only found in proteins. So a polypeptide is a protein that has lots of these peptide bonds. The goal of the central dogma is to take your DNA and turn it into a protein, turn it into something that has lots of those peptide bonds holding it together. For us to be able to do that, we're going to need to do 2 different steps. The first step is called transcription. Transcription is where we take our DNA and we turn it into something called mRNA. The second step is what we call translation, where we take that mRNA and turn it into a protein. As we're going to talk about more today, the process of transcription, when we're doing transcription, we're taking one form of genetic information, which is DNA, and turning it into another form of still genetic information, so mRNA, another set of directions. It's not until we do this process of translation where we take those directions, that genetic information, and turn it into something that can actually do a job. So translation, the way I like to remember this one, is we're translating or going from information into something that actually functions or actually does a job. In transcription, it's all still directions. There's no protein made in transcription. In translation is when we actually start to make a protein. So we're going to talk through both of these steps, transcription and translation, the way that a cell builds proteins. Before we can do transcription or translation, our cell has to have DNA. DNA. Now let me type for us here what DNA stands for, because this is going to be an abbreviation that's important for us. It's deoxyribonucleic acid. Deoxyribonucleic acid. That's what DNA stands for. Now, this nucleic acid part that I see right here, your nucleic acids are your information molecules in the cell. So we've got DNA and we've got RNA that we're going to talk about today. anytime you see nucleic acid, you should be thinking information or directions. This beginning part, deoxyribo on it, this tells me about the kind of sugar that I have inside this information storage molecule. So the deoxy part of the name of deoxyribonucleic acid means we use a type of sugar called deoxyribose. Deoxyribose. So this is the kind of sugar, as you'll see later in the lesson, I ask you what kind of sugar is in the backbone, or what forms the outline of DNA. It's a special kind of sugar called deoxyribose. So when you're looking at DNA, we usually see it in this format you see here called the double helix. So an important idea about DNA, an underlined highlight star idea about DNA, is that DNA has two strands. So DNA is double-stranded, meaning that you're always going to find one strand of DNA attached to another strand of DNA. The fact that DNA is double-stranded makes DNA really stable. So this is a good thing for you. It means your DNA is not going to fall apart. Your DNA is really strong. It's really stable. When we look at these two strands of DNA, we would say that these two strands of DNA are what we call complementary. Complementary. This is another underline highlight star word. Complementary. For my friends who've had a chance to start on this lesson, when we say that the DNA double helix is complementary, does that mean that these two DNA sequences, these two strands that are connected, they are or they are not the same sequence as each other? Do these things match? The letters in each one, do they match? Yeah, they're not the same. Absolutely. When I say that the two strands of DNA in your double helix are complementary, I mean, like Fanchen mentioned, that they are mirror images to each other. They are not identical. So here's some stuff for us to know about complementary. Strands of DNA being complementary, they are not identical to each other. What I mean by that is if I have an A in one strand, I do not have an A in the other strand. However, I can use the sequence of one strand to figure out what the sequence of the other strand would be. So like Fanchen mentioned, we're talking mirror images here. So use the sequence of one to predict the sequence of the other. Complementary strands of DNA. They're not the same. The sequences are not going to be the same. But if you know the sequence of one, you can figure out the sequence of the other one. The way that we can do that is based on the sets of pairs. So DNA bases will always pair together in a particular way. So the first pair of nucleotides, the first set of bases that are always paired together are A, which is adenine, and T, which is thymine. So when I look at my strand of DNA here, notice how I have a thymine in this strand of DNA. In the complementary strand of DNA on the other side, I will always have an adenine. If I go down here, I've got another thymine in this strand of DNA. Across the double helix, I've got an adenine. Or if I've got an adenine in one strand, I'm going to have a thymine in the other strand. When we talk about them being complementary, anytime you see an A, the other strand has a T. And anytime you see a T, the other strand has an A. My other set of bases my other set of letters that I have in DNA are C and G. So anytime, remember we're complementary, right? So anytime I see a C in one of my strands of DNA, the other strand is going to have a G. Or anytime I see a G in one strand, the other one is going to have a C. So if we look over here at our double helix, here is a G in this strand of DNA, and in its complementary DNA, we've got a C. Or over here, we've got a C in the complementary DNA, the other half of the double helix, we've got a G. C and G will always be paired to each other. A and T will always be paired to each other as well. Now, over here on the right, we see a really complicated chemistry picture, and I promise that I will never make you identify these chemical bases based on their shape. So I'm never going to ask you to draw me a thymine, or I'm never going to show you a picture of adenine and ask you to tell me which base it is. That's not why I put these pictures in here. The reason I included this picture is to show you why adenine, or A, always pairs with thymine, T, or why guanine, G, always pairs with cytosine, C. It has to do with the chemical structure of these nucleotides. See over here on adenine, how adenine's got an amine group sticking up here, and it's got a nitrogen right here. Notice that this nitrogen interacts with an amine group over here on the other nucleotide. And this oxygen over here on T interacts with the amine group over here on adenine. There are two places, so two functional things, if you will, where I can form hydrogen bonds to connect these nucleotides together. A and T both have the ability to form two hydrogen bonds. Because they both can form two hydrogen bonds, they will form bonds with each other. When I go down here and look at guanine and I look at cytosine, notice that these nucleotides have the ability to form three hydrogen bonds. So when we're building our double helix, we're always trying to build it so that everybody can build as much hydrogen bonds as they want. A wants to form two, T wants to form two as well. C wants to form three, G does too. So we'll pair these bases together based on their chemical structure. Now, again, I will never ask you to identify a nucleotide based on its chemical structure. I'm never going to ask you to draw a nucleotide's chemical structure. But hopefully by looking at this, we can see those three places that we form those hydrogen bonds and those two places that we form those hydrogen bonds to help us understand which bases form bonds with each other. Now, I kind of have a nerd out fact moment here that I have to share with you all. Dr. Aulis has a favorite nucleotide. Does anyone want to take a guess, or does anyone know, because they listened to other videos, does anyone know what my favorite nucleotide is? We're all nervous to guess. Okay, Gerardo's voting. We got to vote for cytosine. We got to vote for guanine. Random guesses. Yeah, no, it's totally cool. So let me pull here. Let me search. The reason I have a favorite nucleotide is I literally have a cat that is named after a nucleotide. In normal times, and what my daughter calls our cat is Addie. Addie short for adenine. So Dr. Aulis's favorite nucleotide is A, because I literally have a cat named Adenine. No joke. The only time we call her Adenine is when she's naughty, right? That's the only time your mom ever calls you by your full name. But my cat's name is Adenine. right? Kira's like, that's the one that nobody guessed. Yeah, so I've got a cat named Addie. If I ever got another cat, I would probably have to name that cat Minnie, right? Thymine. So they could pair together, right? So I've already got an adenine, need to get me a thymine. I could also get me one called Uri, we're going to talk about later, called uracil. That's an RNA. So got me an adenine. Now I got to figure out getting me a thymine and a uracil, and then I will be the science cat lady. So Dr. Ellis's favorite nucleotide, adenine, for my cat. Right, with that awkward moment when you wanted to get a thymine and you accidentally get a guanine. I bet that if I ever got a cat that didn't integrate well with adenine, I would have to change its name, right? I could call it Cyto, or Guano. That'd be really funny. A cat named Guano. That's what you'd call a bat, right? A bat named Guano. So fun times with Dr. Ellis here. My cat adenine. All right. So when we talk about the genetic information in your cell, the kind that we've just talked about so far is something called deoxyribonucleic acid. So remember, we talked about how the DNA, the deoxyribo is the kind of sugar that's in it. Nucleic acid means that it's information. When we're talking about the central dogma, we not only take deoxyribonucleic acid, we not only use that, we also use what's called ribonucleic acid. Ribonucleic acid, instead of using deoxyribose sugar, it's going to use something much simpler. Called ribose sugar. So sorry, y'all. It did its daily, we've got a crash in the middle of class. I think I caught it before I kept talking. So we're back in business. We mentioned before my computer died that deoxyribonucleic acid, that's our DNA. We start with that. A cell always has that. When we're trying to build proteins, we're also then going to use something called ribonucleic acid, or RNA. The difference between deoxyribonucleic acid and ribonucleic acid is the kind of sugar that I use to build it. So where over here we had deoxyribose sugar, that's what its name means. Over here we have what's called ribose sugar. Ribose sugar. The thing when we talk about using ribose sugar to build our genetic information made with ribose sugar tends to be single-stranded. So notice in my picture over here, I've got a molecule of what we call mRNA that's involved in the central dogma. It's made out of a backbone that has ribose sugar in it, instead of what we saw over here being deoxyribose sugar. The only difference, by the way, between ribose sugar and deoxyribose sugar is deoxyribose has one less oxygen. That's what the oxy part means, oxygen. So if we went down nitty gritty at the chemistry level, which I'm not gonna make you draw any kind of sugar. If we get down to that level, Deoxyribose is a sugar that has one less oxygen than ribose sugar. And in general, that's not really a problem when it comes to our nucleotides that we use. They play nice, if you will, with ribose sugar, just like they do with deoxyribose sugar, except for this one right here, except for thymine. So thymine is a little bit particular about the kind of sugar that it's willing to attach to. Thymine is like, yeah, no, not like in the ribose. When we build molecules of RNA, when we're building something that has ribose sugar, we're not allowed to add the nucleotide base thymine because thymine is too cool for ribose sugar. In the place of thymine, everywhere where we would have put a T into our DNA or RNA molecule, we have to use a new base, and that new base is this one right here. The new base is uracil. So this base uracil is only seen in RNA, only in RNA, and it takes the place of thymine. It's going to sub in everywhere that we would have put a thymine. We're now going to put a uracil in its place. By the way, all this talking that we're doing, when I talk about building RNA, RNA is also complementary to DNA. When I say that RNA is complementary to DNA, remind me, that means it is or is not identical. If RNA is complementary to DNA, it is or is not identical. Yeah, exactly. Complementary, remember, means we are not identical. Not identical. I can use the sequence of one to predict the sequence of the other. So when I use DNA to build RNA, it's not going to look exactly the same. It's going to look a little bit different. Just like when I take one strand of DNA and I use it to build another strand, that's not identical either. Let me check something really fast. I'm going to go a little bit out of order because I want us to practice some of the stuff that we just said. So I'm going to kill our group work activity. I'm sorry. We'll do it together as a class. We can practice again. I'll build us a new one for next week. For those of you that snuck ahead in the group work, you saw me asking you to build complementary DNA. And I gave you the sequence that you see right here. When I talk about complementary DNA, again, it's going to be the exact same question. Complementary DNA, does that mean that it is or is not going to be identical? I've got one strand of DNA right here. When I build its complement, is that going to match? Yeah, it's not going to match, right? It's going to be the mirror image. It's going to be the opposite. So my first strand of DNA that I see right here, I've got a T, which means what am I going to put in my complementary DNA if my first strand has a T? Yeah, if I've got a T in one strand, I'm going to put an A in the other one. Absolutely. And my next base is an A. So what am I going to put in the other strand? Yeah, now I'm going to put a T. Okay, and then my next base is a C. That's a new one for us. If I've got a C, exactly, I put in a G, and then I've got a G, so I'm going to put in a C. Okay, so we can work our way all the way through this. I probably won't do all of it, but we can work our way all the way through. The idea of building complementary DNA. You use the sequence of one strand to predict the sequence of the other It is never identical. They don't match exactly, but we know who pairs with who to help us build that molecule. Now check this out. I asked you to build the complementary DNA sequence. That's one kind of question you'll see on the homework assignment. The other kind of question you'll see on the homework assignment is when I ask you, I might phrase it, that I ask you to build me complementary mRNA. Complementary mRNA. The process of building complementary mRNA is a process called transcription. So I can ask this question two ways. I can ask you to build me complementary mRNA, or I can ask you to do transcription. When I talk about building complementary mRNA or doing transcription, you do the exact same thing that we did on the previous slide, except there was one base that I can't use when I build an mRNA molecule. Which of those bases doesn't play nice with ribose sugar? Who's too cool for ribose sugar? Exactly. Thymine is too cool for ribose sugar. What letter in my DNA sequence would tell me normally that I would need to put a thymine down here? Who normally pairs with thymine? Yeah, exactly. My A's, it's not normally, it's always, my A's always pair with T's. But in the case of building RNA, I can't put a T with an A. Now I have to use that special base that we just talked about, right? Now I've got to use that uracil. So here's a fun fact, or kind of a workaround for you. You can build a complementary mRNA sequence exactly the same way that you built your DNA sequence. All you have to do is trade out any of those T's. So remember in this one, when we did DNA, it would have been a T. Well, now that I'm doing RNA, I'm going to put a U instead. So when you're working on an assignment, if I tell you first to make the DNA and then I tell you to make the RNA, good news, you can use that DNA that you made, your complementary DNA, and just sub out U's for those T's. So the only difference between complementary mRNA and complementary DNA is whether or not we use U, whether or not we're using uracil. I'm pretty sure, by the way, that you have this exact sequence in your lecture notes to do the processes of transcription and translation on. So I don't expect you to have caught all of them already. You can definitely practice building these things again. Let me go back to where we were. We were talking about RNA. And I'll mention that when we're talking about RNA, there are actually 3 different types of RNA that we see. So the one we've talked about so far is the one that's called mRNA. mRNA stands for messenger RNA. mRNA is really important for us to get our message of what protein we want to make from one place in the cell to another place in the cell. To help us figure out what those places are, let's start with this question. Let's take a step back. When we talk about the genetic information in your cell, the genetic information in your cell is stored as DNA. When we talk about DNA, where would we say in the cell that we find your DNA? Where is that DNA located? Yeah, exactly. DNA is located inside the nucleus. So DNA is located inside the nucleus. Do you remember from lab which which kind of organelles we said really helped to build proteins? Who really helps us to build proteins? Yeah, so the main one that builds proteins are those ribosomes. Ribosomes are the big one. And then we also use that organelle called the rough ER, the rough endoplasmic reticulum. Endoplasmic reticulum, the rough ER. Both of those build proteins. Hey, I promise this is not a trick question. Ribosomes and the rough ER, are these things located inside or outside of the nucleus? Ribosomes and the rough ER, do you find them inside the nucleus or outside the nucleus? Yeah, I promise it's not a trick question. It sounded tricky, but it's not. Inside the nucleus, I only find the DNA and I find that little dot called the nucleolus. I store your DNA in the nucleus to keep it safe, to make sure that the sequence doesn't change, all that kind of stuff. But when I want to use that DNA to build proteins, my protein building machines, the ribosomes and the rough ER, they're not inside of the nucleus. So I need to get my genetic information, the kind of instructions that are found in the DNA, I have to get them out of the nucleus so that I'll be able to build proteins with them. The way that I get my information out of the nucleus to these organelles that can actually use it to build proteins is by using this molecule called mRNA. So messenger RNA, its name is a really great representation for us. mRNA, messenger RNA, takes the message out of the nucleus. The job of messenger RNA is to take the message of how to build a protein and get it out of the nucleus. That's mRNA's job. When I get that message out of the nucleus, my goal with it is to turn it into a protein. Or my goal is to build a long chain of amino acids. So amino acids are we have to take a message that's in the form of nucleic acids, and I have to turn that into amino acids, which is a completely different language, the language of proteins. To do that, I use my next kind of RNA that's called transfer RNA. Technically, it's called transfer RNA. But the way I think that might be easier to remember it, tRNA, I like to call this one translator RNA. Translator RNA. Because if you think about a translator, that's someone who's bilingual. Maybe they speak English and Spanish, for example, a translator, or they speak English and sign language. One language in your cell is nucleic acids. The other language in your cell is amino acids, what I use to build proteins. So my tRNA molecules, what I like to call the translator RNA molecules, these are the ones that speak nucleic acids, they speak directions, and they speak amino acids, they speak protein. For you to be able to build proteins, we need somebody who can go between those two languages. That's the job of tRNA, to take a message and turn it into a protein, tRNA. There is one other kind of RNA that we're not going to really talk a lot about in our class, but there's one last kind of RNA called rRNA. That's ribosomal RNA. Ribosomal RNA is found inside your ribosomes. So your ribosomes actually have a little bit of this inside of them as well. This is going to help your cells to do the process of translation correctly. So one last kind of RNA, ribosomal RNA, that I find inside those ribosomes. I've talked for a long time. I need to pause for a moment. What do we think about these three forms of RNA? Thumbs up, thumbs down, questions. Yeah, I like Fanshin's comment. She mentioned that you can think about transfer RNA kind of like a converter. That's a great way to think about it. Converting from RNA, so from nucleic acids into those amino acids. I like that. Yeah, converter. Go for it, Tammy Teo.
Can you come again with the ribosome RNA, please?
Yeah, so this is the one that we don't care as much about. I just mentioned it to say that it exists. When we talk about ribosomal RNA, rRNA, it's found actually inside your ribosomes. So when we're doing the process of translation, when we're taking our message and turning it into a protein, we use the ribosomes to do that. But they have a little bit of RNA in them as well to help us, essentially, to help us put our mRNA message in the right place inside of them. So I'm trying to think of a good analogy. It's kind of like when you're building furniture and they've like pre-drilled holes for you, for example. So those pieces fit together well and you can just screw into them. Kind of think about our RNA like that. So we've kind of like drilled some holes or made some spaces inside the ribosome just to make sure that our mRNA comes in to the right place so we can read it. So basically to give the shape to the ribosome. Maybe that would be a good way to think about it. In our class, when we're talking about the process of translation, we're really just going to care about tRNA and mRNA. I'm going to mention that this exists, but we really just care about tRNA and mRNA. We're discriminating against it. Poor rRNA. Okay, so let's well, first, before I move on, I gotta toss up my cheesy humor here. So let's figure out why this cartoon is funny. Here is a nice molecule of dRNA. I know it's dRNA because I've got a double helix. There's two strands. We've got two mRNA molecules here in the front. And this mRNA molecule, Arthur, says, I'm telling you, Carol, I'm done being this guy's messenger boy. He can leave the nucleus and do it for himself for all I care. What does this cartoon say that is biologically accurate? What are some of the facts that we see in this cartoon that we just talked about? Uh-oh, we're nervous. Okay, perfect. I love it. So first thing we're honing on is messenger boy, right? mRNA, that's definitely mRNA is my messenger RNA. Absolutely. Okay. What about this one right here? Yeah, exactly. Like Anisha said for us, the DNA is in the nucleus. DNA is in the nucleus. Absolutely. So DNA can't leave the nucleus. That's what keeps it safe. So DNA can't leave the nucleus. It's stuck inside of there. mRNA is my messenger. That's what allows us to get information out. Now, we don't usually think about mRNA as being disgruntled, right? The disgruntled employee, but this one, man, he's at the end of his strand. By the way, mRNA, it can leave the nucleus, but it's single-stranded. Single-stranded things don't survive very long. So DNA is really stable because it is a double helix, but it also can't leave the nucleus. We make something that's single-stranded, that's one strand, number one, so it can fit out of the nucleus, so it's not stuck inside. But number two, because we can pick what kind of information we put into it. I should have included a little bit of information in here about this. Does anyone know, with the Moderna vaccine and the Pfizer vaccine, how do those vaccines for COVID relate to what we're talking about? Does anyone know? Yeah, Fanshin's right. The Pfizer vaccine and the Moderna vaccine, they are examples of mRNA vaccines. The same kind of mRNA that you and I are talking about, that's normally in your cells. mRNA is instructions for what your cells should build. When mRNA is in a cell, ribosomes use it to build proteins. But here's an important idea that goes along with some of the vaccine skepticism right now about these vaccines. mRNA never goes into the nucleus. mRNA never actually interacts with DNA. In your cells, normally, you'll build mRNA based on your DNA, and you'll shuttle it out of the nucleus. That mRNA never comes back in to the nucleus. And actually, it won't live very long in your cell before we break it down, before it falls apart, because it's just single-stranded. When we talk about these vaccines, we take some of this messenger RNA, send it into your cell, your ribosomes use it to make directions to build proteins that we would normally see built on the outside of COVID-19 or the coronavirus, excuse me. We build those proteins, we send them outside of our cell, and our immune system responds to them. But what's never going to happen is the mRNA from those vaccines is never going to go in and interact with your DNA. All our cell ever does is it uses mRNA to build proteins. Then we spit those proteins out into our cytosol. If they're flagged where they're supposed to go to the surface of the cell, they'll go to the surface of the cell. But mRNA never impacts your DNA sequence. It's not going to change your DNA. It's just going to change what proteins your cell's building for a little while. Fanshin asked a question about denatured proteins. It's not going to cause you to build denatured proteins because it's building a very specific shape of proteins called the spike proteins. Most proteins that you'll ever bump into your life are not denatured, and they don't have the ability to change any other proteins in your body. And the spike protein is that way, where it's not going to change any of your normal proteins. It's going to look like its name suggests, it's going to look kind of like a spike. So it won't denature any of your proteins. And those proteins that we built are only going to float around long enough for your immune system to find them all and cause a reaction to them. So not going to denature your proteins, but a good question though, good correlation. Excuse me. So today, after class, you can now say that you better understand the scientific basis for those mRNA vaccines, getting the message or the directions to build a particular protein. The protein that we're talking about being the spike protein, that thing that lives on the outside of the coronavirus. So let's talk about how the process of building a protein works, how the central dogma actually goes. So here's how it goes. And I'm gonna, since we all care about it right now and it's important to us, I'm gonna use the example of coronavirus here. So inside the genome, of coronavirus, this virus that infects us, it has genes, and our DNA has genes too. So our genes are for things like the color of your eyes, or the texture of your hair, or how to build your keratin protein. We'll talk about keratin next chapter. We have genes, and the thing for you to know about genes is that they have the specific directions for a particular protein that you're trying to make. See, every single cell in your body has the directions in your DNA to make every single protein you could possibly need. So from the keratin proteins that we have in the skin outside of our body, to the cytoskeleton proteins that neurons use to have that funky shape, every single protein you could ever need to build in any of your cells, everybody has the directions for that. But everybody's not building those proteins. Your neurons are never going to use the gene to make hemoglobin. They're never going to build hemoglobin. But they are going to use those genes for building neurotransmitters, for example. So we've got a gene, its directions for how to build a particular protein. And again, let's go back to coronavirus. Here's my gene for the spike protein. That's a protein that lives on the outside of the virus. Ahead of the gene, there's a place called the promoter region. Think about the promoter region kind of like one of those billboards on the side of the road. Now, just for my own knowledge, I'm curious to know, was anyone who's here in class today driving before they had a smartphone? Was anyone driving before they had a smartphone? I was. Okay, Tien was. At least one other friend who's, okay, perfect. Okay, I got lots of us. Excellent. Usually when I ask this in class with students in person, I have very few people who were driving before they had a smartphone. It's like, oh, you all were lucky. we had to print our directions, and we had to like know where we were going. I say that to say back in the day, if you didn't have a smartphone, yeah, when you had to use MapQuest, right? When I didn't have a smartphone and I was driving down the highway and I got hungry, I had to look for billboards to know what was nearby. Unless I happened to do a search before I left to know where things were, I had to watch those billboards to know where I could find. I'm an Okie, so where I could find my Brahms. I do love me some Brahms. So I'm driving down the highway, I'm feeling ice cream, and all of a sudden I see a billboard for Brahms. It says 2 miles ahead. I'm like, score! I finally know where I can get my ice cream. Think about that like what the promoter region is. The promoter region says to your cell, hey, over here is where you'll find the directions for hemoglobin. So if your cell wants to make hemoglobin, it's looking for that billboard for hemoglobin. Or in another place in your DNA, the promoter region is going to say, hey, over here is where we have the gene for keratin. And if your cell wants to make keratin, it will use that promoter region to find it. So every single gene in your body has this place that's in front of it called the promoter region. This is how your body knows where your genes are. So how it knows where it should go to build that particular protein. So the reason I need a promoter region is we've got this enzyme in your cell called RNA polymerase. RNA polymerase. Its job in life, this polymerase word means we're building something. This RNA part right here means we're building RNA. The one job of this enzyme right here is to build you RNA. But it's kind of like those of us who were driving before we had a smartphone. We don't necessarily know where in the DNA each of the genes are that we're looking for. So we look for those billboards, and this promoter region, that billboard, tells RNA polymerase that this is where it should attach to your DNA. So when you're thinking about the function of the promoter region of your DNA, let me back up. When we're trying to figure out what the function is of this promoter region, this is the place where my enzyme RNA polymerase can attach to my genetic information. So I need to have this location, this promoter, in front of my gene to give me a place for the enzyme to attach. If for some reason, I was talking about Oklahoma, right? So if for some reason in Oklahoma, a tornado comes through, not all that uncommon, we blow away that billboard. If I didn't have a smartphone, I wouldn't know that Brahms was 2 miles ahead and I would drive past it. If I don't have the promoter region in front of a gene, RNA polymerase is going to look around. It's hungry, if you will, or it's really interested in making hemoglobin, but it's not going to be able to do that because it can't find the place to attach. So the promoter region tells RNA polymerase where to attach, where it should bind to your DNA to help us get those directions. So let's say that we found that billboard, that RNA polymerase has come and attached to the double helix, your DNA. It opens up your DNA. We go from having 2 strands to being open. Now RNA polymerase is going to go through and read the sequence of your entire gene. So this is my gene for the spike protein, if we're talking about coronavirus, or this is the gene for keratin, if we're talking about skin cells. RNA polymerase starts at the promoter region and goes through and reads the sequence of this gene, 1 base at a time. And while it reads it, it makes complementary RNA. So it takes the sequence of the DNA strand and it builds the complementary RNA that would match it. Again, my favorite question for the day, complementary means it is or is not the same. Complementary is or is not the same. Yeah, exactly. Complementary is not the same. So RNA polymerase is going to go down and one by one, it's going to read the sequence of this DNA molecule and it's going to build an RNA molecule that's the mirror image to the DNA. So we end up with something like this. Here's my DNA. I had a thymine and an adenine and a cytosine and a thymine, adenine, and guanine, etc. RNA polymerase goes down one by one. I had a thymine in the DNA, so I'm going to put an adenine in my RNA. I had an adenine in my DNA, so I'm going to put a uracil in my RNA. Hey, remind me, uracil is going in instead of which nucleotide? Which one do I not have in RNA? Yeah, exactly. I do not have T. So there's no T's going in. Normally it would be A would go with a T. But when I'm building RNA, I put in a U instead. And now I read here, I've got a C in the DNA. So I put a G in my RNA. RNA polymerase goes down and reads the entire gene all the way down that gene to make a copy of that information. A copy in the form of RNA. The reason I have to do transcription, the reason I have to build this RNA is so that I can send my message out of the nucleus. I've got to send that message to where it can actually be used. And where it can be used is those ribosomes and the rough ER. Those are the places I can use it. Before we talk about our analogy for the process of transcription, I want us to work together on this table from your notes. Can you help me out in the chat? What page of your notes packet do you have this table? Where do we have this? Okay, I'm hearing page 5. Perfect, page 5. So let's go through together. We're going to compare the two types of genetic information that we've just talked about, just to make sure we have these things straight. And then we'll look at an analogy for transcription, turning DNA into RNA. Let's start with our first row on the table. The first row on the table asks us where in the cell we would find it. We said DNA is only ever found in one place. DNA is always, always inside the nucleus. Always, 100% of the time. The only time it's not inside the nucleus is when we're doing mitosis, right? When we do mitosis, the nucleus disappears and then the DNA is not in it anymore. But otherwise, DNA is always in the nucleus. When we talk about RNA and we're thinking about its location, this one is maybe a little bit trickier to describe, but RNA, yeah, so multiple of us are saying it's both inside and outside the nucleus. So I'm going to expand on that. I'm going to say it starts in the nucleus and then it moves to the cytosol. It moves to the cytosol. So it's both inside and outside of the nucleus, right? We build it in the nucleus where I have my DNA, and it goes to the cytosol, which is the soupy stuff outside the nucleus. This is your key with RNA. This is the reason I have to have RNA, because I need to get this message into the cytosol. I've got to get it out of the nucleus. Let's talk about some of the basics of DNA and RNA. Does anyone remember the name of the sugar in DNA? It's a long word. I'm making you type a long one. Yeah, exactly. The long name of the sugar that I find inside DNA, it's called deoxyribose sugar. That's the D, right? Deoxyribose. RNA is way better, right? It's a lot shorter. What does RNA stand for? Yeah, there we go. RNA is just ribose sugar. Ribose sugar. When I have the differences in their names, I have a different kind of sugar. I also have different nucleotides. Let's start with the 3 nucleotides that DNA and RNA have. What nucleotides do DNA and RNA both have? What do they both have? Perfect. They both have A, C, and G. When we're talking about RNA, what's the other one RNA has? Yeah, RNA also has U, also has uracil. Instead of uracil, DNA has what? What does DNA have instead of uracil? Exactly, thymine, T. Something that will never show up on an assignment or an exam. Who remembers Dr. Ellis's favorite? Which one is my favorite? That's right, adenine. A is my favorite. My very energetic cat, adenine. That one's my favorite. But A, T, G, and C, all of those in DNA. RNA, A, U, G, and C. Last question, single-stranded and double-stranded. Which one of these is single-stranded? Who did we say was single-stranded, DNA or RNA? Who's single-stranded? Yep, RNA is single-stranded, absolutely. And DNA, what makes it so stable is the fact that it's double-stranded, double-stranded. This is an underline, highlight, star, must know table. We need to be able to compare and contrast DNA to RNA. I promise you it's going to show up on the homework, and I promise you it's going to show up on the exam. So make sure we know the similarities and the differences between DNA and RNA. Hey, by the way, I will mention something for you too. This difference between having uracil and thymine, this is probably the most important difference between DNA and RNA. I say that because when you're working on the homework assignments, anytime you see a list of letters that includes T, you're always talking about DNA. DNA is the only one that has T in it. Anytime you see a list of letters that has U, you always know you're talking about RNA. RNA is the only one that has U. So pay attention to those T's and U's. If I ask you on the homework assignment to build me complementary RNA, you better not give me any T's. T's are wrong. If I ask you to build complementary DNA, do not give me any U's, because U's are wrong. So that's a big one that helps us know what kind of genetic information we're looking at, and helps us to be able to build our genetic information correctly. When we talk about the process of transcription, here's an analogy that I want to give you for it. If this analogy helps you, awesome. If it doesn't help you, just forget I even said it. But when we talk about the process of transcription, where we take your DNA instructions and turn them into RNA that's very specific with the information you want, I like to think about this kind of like what we used to do in the olden days, as my family called it, when we didn't have smartphones, when we didn't have the internet. In the olden days, when you were doing a project for school and you needed to do a little bit of research, you'd go to the library, and the library had this great set of something called Encyclopedia Britannica. I loved this as a child, Encyclopedia Britannica. This was basically like what used to be what the internet is now. All the important information inside one set of encyclopedias, it's all there for you. So Encyclopedia Britannica, from A to Z, all kinds of topics, all kinds of information you could want to know about. So I mentioned this in the guided lesson. I'm pretty sure I mentioned this in the guided lesson. Man, when I was in elementary school, I was super interested in aliens. So hanging out in the A's with those aliens. Even though I lived in Colorado, I was also strangely drawn to tornadoes. I guess that was foreshadowing for my life in Oklahoma. So depending on what topic you're trying to learn about, you would pick up a different copy or a different index, I don't know what you call them, a different book out of Encyclopedia Britannica. So if I'm interested in learning more about tornadoes, I'm going to pull out T. I'm not going to pull out A. Just like in your cells, if I'm really interested in making the hemoglobin protein, I'm not going to go and pick up the instructions for the book that talks about keratin. So your cell has all of these encyclopedias. It has your DNA that has all the directions for everything you could ever need. Your cell knows that it wants to make one particular protein. Just like I know, I'm really interested in learning more about tornadoes. So I'm going to go to the book that is flagged for tornadoes. I go about halfway through that book. I see the page about tornadoes. Awesome. Third grade Dr. Ollis is nerding out. Oh my gosh, this is so cool. But here's the thing with Encyclopedia Britannica. Back in the day, you couldn't check it out from the library. The library kept its Encyclopedia Britannica at all times, kind of like how your cell keeps your DNA at all times. If I want to take that information with me, I definitely don't have a smartphone, because this is third grade Dr. Ollis, man. I wasn't quite wearing this kind of clothes, but close. not quite. Yeah, if I want to take that information with me, I am going to have to pull out the copier. I'm going to have to put in my nickels into the copier, and I'm going to have to make a copy of the information I want. But if I'm paying a nickel a page, I don't want to copy all kinds of extra information, right? My allowance only goes so far, man. My $1 a week, not really. Let's be real, I didn't actually get an allowance. It was really sad. But my nickels, they're limited. They only go so far. So I'm only going to copy the pages that actually have the information I want. Just like in your cell, they're only going to make mRNA with the information that they actually want. So your cells go in, they copy the instructions for hemoglobin. They get their part that spits out with the hemoglobin directions, and they take that out of the library. Just like I go in and find my encyclopedia, find my page about tornadoes, copy it, and I can take that with me. So there are several parts of this analogy that I ask you to pair up with parts of the cell. I did not put that on my slide. For those of you that have your notes in front of you, can you list for me the things that I told you I wanted you to try to relate to the cell? I want to make a list and then we'll relate them together. I know that the encyclopedias was something that I wanted you to relate to a cell. The copy machine, okay, copy machine, the copies. Did I have you? Okay, I was gonna say, I was thinking I had you do the library too. Okay, is that everything or were there any others? That might be everything. Okay, so help me out. We've just talked through this analogy a whole bunch. When I talk about the library, what might we say in your cell that the library represents? Any ideas? Yeah, exactly. Krista and Fanchon are chiming in. The library represents the nucleus, the part of your cell that is sacred, if you will, the part of your cell where we store the encyclopedias. The encyclopedias are representing something that can't leave the library. What can't leave the nucleus? Yeah, exactly. DNA cannot leave the nucleus. All of that knowledge in those encyclopedias, it's not leaving the library. I'm going to have to find a way to get that information out of the library. Well, the way that I do it is I make copies. What do the copies represent in the process of transcription? What do those copies represent? Yeah, exactly. The copies are going to represent the RNA. They're going to represent the information that leaves the nucleus, that goes into the cell. Carissa is totally right, though, when she talks about RNA polymerase. RNA polymerase is like my copy machine. RNA polymerase is what I can use to take that encyclopedia with all of its information and make my copies out of it. Just like in your cell, RNA polymerase takes your DNA and it copies or it builds for you RNA. Because remember, RNA can leave the nucleus, DNA cannot. So as you're studying the process of transcription, think about Dr. Aulis in her childhood library, not quite dressed like this, using a copy machine, maybe a little bit better than this one, with those little nickels, copying my information about tornadoes and aliens. I also really liked ancient Egypt. I'm not sure if I mentioned that in the guided lesson. So Dr. Aulis at the library. If it helps you with transcription, awesome. If it doesn't help you with transcription, that's okay too. How do we feel about transcription? Going from DNA to RNA. Thumbs up, thumbs down, or some other random emoji. All emojis welcome. Here's our daily penguin emoji. Remind me at the end, I'll try to draw you another one. I like it, Kaylin. Very emotive. I don't know if that's actually a word, but I like to pretend it is. All right. Well, we've got about 20 minutes left. And in this time, I want to introduce you to translation. I think, Carissa, I think it was you that emailed me about wanting to talk about how to use that table. So I think we're going to have just enough time today to talk about how to use that weird table that you have in your notes. So we're going to talk now about the process of translation. So let me back up to remind us where we're at. So remember, this is the central dogma. The central dogma is how I take DNA, turn it into a protein. What we've done so far is we've taken the DNA and turned it into RNA. We've done transcription. Now that RNA is leaving the nucleus. It's going out to ribosomes. Am I still cutting out or am I back? Because I heard it be funky, but I think I'm back. Maybe I'm not. Check, check. Can we hear me? Okay, we think I'm back. Let me re-say what I said before, just in case we missed it. So far, we've talked about transcription. So far, we've talked about taking DNA and turning it into mRNA. Now, I've got to do the hard work. Now, I've got to turn that RNA into a protein. I actually have to build my protein now. I need to do translation. So at this point, my mRNA message has left the nucleus. My DNA is still safe. That mRNA went outside. Now I'm going to use it to build a protein. So the way the process of translation works is I take my mRNA message that has those specific directions for the protein I want to build, and I bring it over to a ribosome. Remember, ribosomes are the small organelles that are floating around in your cell. They're able to take those messages and turn them into a protein. So my mRNA message comes over to a ribosome and it goes inside the ribosome. The ribosome is going to go through and read this message, and it's going to read it in little small pieces. So the way that we read messages is we use something called codons. A codon, that's an underline highlight star word. We've got to know codons. Codons are sets of 3 nucleotides. I like to talk about ribosomes kind of like early readers. They're learning how to read. They can't read more than 3 letters at a time. Now, I probably won't have any takers on this, but when anyone was younger or with your younger siblings, did anyone ever use the Bob books? They had little red ones, little blue ones. I think the yellow ones were like the advanced Bob books. Okay, at least one person knows what I'm talking about. Bob books are books that like literally all the words are like 3 letters or less. So you get a story about like Bob and Pat. So I can never come up with like a story with 3 letters words, but like Bob and Pat see a cat. Bob and Pat go, I don't know. I can't come up with another one. Get a hat. There we go. I like that. Bob and Pat get a hat. Oh, look at you all. Like y'all are great at writing Bob books. Bob and Pat go to the zoo. It's totally true that Bob and Pat would sit on a cat. Absolutely. So big idea. Oh, nice. Carissa says they've got them at Costco. So if you want to relearn how to read, you can go get you some Bob books at Costco. Just like the Bob books have really short words in them, your ribosomes can only read short words too. They can only read these codons. So mRNA comes into the ribosome. The ribosome sees a set of 3 letters. It sees a codon. And the ribosome calls out to the environment next to it, hey, AUG. I'm reading the letters AUG. Can anyone tell me what amino acid AUG stands for? And floating around next to this ribosome are our tRNA molecules, our translator RNA molecules. So ribosome calls out AUG, and this translator over here says, oh yeah, I know what AUG means. Totally right in the chat. It means methionine. That's the amino acid that you're supposed to add when it says AUG. So the tRNA that can read or can translate the set of 3 letters that are in the RNA, that tRNA comes into the ribosome, and the ribosome says, sweet, we've read this codon. We've read this set of 3 letters. Now it goes next door. CUG calls out to the environment. CUG. Who knows what CUG means? And the translator that can translate or can read CUG comes over with its amino acid. As these tRNAs with their amino acids come in, we start to build a long chain of these amino acids. So this tRNA molecule, it came into the ribosome when this set of bases was over here, a little bit farther to the right. But as soon as the ribosome read this set of 3 bases, it actually shifts them over to the next position. The tRNA is still there with its amino acid. So then it reads this base and it shifts the genetic information over again so that there's a new set, a new codon for it to read. So we're constantly moving these tRNA molecules. They're moving from from right to left, working their way through the ribosome. And their amino acids, which is what we were trying to connect anyway, their amino acids are building a chain that actually comes out of the ribosome. So this process goes on and on. The ribosome moves all the way down this mRNA molecule, and it keeps talking to those translators in the environment. It keeps asking them, which amino acid are we looking for? And those translators keep bringing those amino acids over, and we keep adding them to the chain. This is the process of translation. So using the sequence in the mRNA to bring over the correct amino acids. Now this process goes on all the way through this mRNA molecule. So again, we can only read it in sets of 3 bases. Ribosomes are really simple. They only read those codons. Every single protein that we make, so every single mRNA molecule that your ribosomes read, always start with AUG. AUG, that's the codon that we always start with. My tRNA, my translator that reads that, brings over the amino acid methionine. Now in this particular mRNA strand, then my next codon was CCA. So CCA, that's different from AUG, which means a different translator with a different amino acid is going to come over to help us read that. And then my next codon, UCG. UCG, a different translator, is going to come over and read that, and it's serine. So my ribosome goes codon by codon. It calls out the sequence that it sees, and the correct nucleotide, or excuse me, the correct tRNA molecule always comes by. But then we have some things that are called stop codons. And I like to use that analogy of calling out, because the ribosome calls out when it hits a stop codon like UGA. The ribosome calls out UGA. Who can tell me what UGA means? And there are no translators that can tell it what UGA means. So it calls out again and says, UGA. What amino acid do I need? What does this mean? And nobody answers. It gets to the point that the ribosome has called out, if you will, to the environment so many times and nobody's translating it, that the ribosome just can't even. It freaks out and it literally falls apart. Your ribosomes are made of two pieces. So if we get to a stop codon, or we get to a set of 3 letters that nobody can tell the ribosome what it means, the ribosome will literally fall apart, and that is the end of the process of translation. So whenever we hit a stop codon, we're done reading the message, we're done building a protein, that's the end of the line. So anytime we have a stop codon, we stop translation. Even if we have some bases in the mRNA message that are after it, we're not going to read them because we get stuck on this stop codon. Nobody tells me what it means. That's the end. So all of our proteins are done being made when we hit a stop codon. When you are working like ribosomes do to build a protein, you can't call out to tRNAs. Alas, we are not surrounded by tRNA, so we don't have those to help us do the process of translation. What we have to help us, though, is something called the genetic code table. The genetic code table. Now, let me make something abundantly clear here as we see this table for the first time. Absolutely do not memorize this table. 100%. Do not memorize any of it, please. You've got way too much other important information to work on. I'm going to give you this table on the homework assignments and on the exam when I ask you to do the process of translation. So you don't need to memorize it. What you do need to do is know how to use it. So let's talk about how we use it. When we look at the genetic code table, notice that this code table has codons on it, sets of 3 letters. So these codons are the sequence that you would see in the mRNA molecule. So the mRNA molecule says not that one, we're over here, says AUG. That's the mRNA, the codon in the mRNA. That's my start codon, methionine. Now, AUG is all nice and color-coded red. Every other codon that you're going to use is not color-coded in bold and all that fun stuff. So we need to figure out how to use this table to find the correct amino acid. So the way this table works is over here on the left, we see 4 options right here. These options represent the first letter of the codon. So I'm totally going to make something up here, make us up a strand, a DNA strand, or RNA strand, excuse me. AUG. Okay, here's my totally made up sequence that I made up on the fly, so hopefully it works. Anytime you have an RNA sequence, the first thing you're going to want to do for the process of translation is split it up into codons. Remind me in the chat, how many letters are in a codon? How many letters in a codon? Yeah, 3 letters. So I'm going to go 1, 2, 3, line. 1, 2, 3, line. Here are my codons. Now, for each of these codons, I want to figure out which amino acid the table tells me I would add. So, AUG, my first letter in this codon is A, so I know I'm in this row. First letter tells me which row I'm in. Now I go to the top. The top is for my second letter. It's AU, so I know I'm in this column. So we're in this row. We're in this column. Now at this point, there's only 4 options in my box. You can either just eyeball it and find it in the box, or if you look over here on the last side, the third letter, it actually shows you we're G, so we're in the bottom row. AUG, this codon leads to the incorporation of methionine. So this I would put in MET. My first amino acid that this chain is calling for is met. Now we go on to our second codon here. I start over at the beginning, just like I did before. My codon is CUA. So we've got row C, we're in this second row here, column U, letter A. The second amino acid I'm going to put into my chain is leucine. So I'm going to put a little leucine here. That's my second amino acid. My third codon, GCC. So we go down here, we're in the G row, the bottom row. We're in the second column because of that C, and it's a C, GCC. I'm going to add alanine, alanine. So the way you use this table is by taking the sequence of your code and going part by part. So whatever the first letter is in the codon, that's going to tell you which row you're going to use. Whatever the second letter in the codon is, that's going to tell you which column that we're using going down. And that'll help you find the box that you're in to find your exact codon. This table shows us something important, by the way. So the last thing we're going to talk about today is the fact that this table is what we call degenerate. Degenerate. That is an underline, highlight, star, must know word, degenerate. What this word degenerate means is that sometimes more than one codon helps me to add the correct amino acid. So our codon, for example, was CUA. But if I had instead used CUC, if that's what my codon said, or CUU, or even CUG, I would still always get the right amino acid. The idea of the genetic code being degenerate is a good thing for you because it means you could still have some mistakes in the process of transcription or translation, and you might still end up with the correct protein. You may still be good. So the genetic code being degenerate is great news for you because it means you're more likely to make the right protein. Now, the fact that it's degenerate is because we have something in the genetic code I am so sorry, guys. Yeah, actually, I think that time was on me. I think I somehow hit some button to close it. So that time was on me. I didn't say anything though. So I know you were hanging on the edge of your seats. What makes it degenerate? Let me tell you what makes it degenerate. What makes the genetic code degenerate is something that we call the wobble base. The wobble base. So the wobble base is the last letter in the codon. The last letter in the codon in all parts of this box, see how no matter what that wobble base was, I still always ended up with proline. Or no matter what that wobble base is in this box, I still end up with threonine. The wobble base is the last letter in the codon. This is the only one that I can change and be guaranteed to get the right amino acid. Because I can do ACU, or I can do ACC, or ACA, ACG. All of these lead to threonine. But maybe if I was supposed to have ACU, but instead I have AAU, Notice how I changed my second letter and I changed to a different amino acid. Or maybe it's not even the second letter that I change. Maybe instead of ACU, maybe I change the first letter. Maybe I've got CCU. If I change anything except for that wobble base, I'm probably not going to get the correct amino acid. So the genetic code being degenerate, it means that there's more than one sequence, more than one codon that still leads to the correct amino acid. But the differences in those codons, it comes down to this wobble base. It comes down to the last base in the codon. We are out of time for today. What I want to remind you of is the fact that there is a transcription and translation practice worksheet that is underneath the guided lesson for lesson number 6. Please print out that worksheet and start working on it between now and next week, just so that you have plenty of time to use the ideas that we worked on in class today. So again, that transcription and translation practice sheet, that's going to help you practice doing some of the kinds of things that I'm going to ask you to do on the lesson number 6 homework assignment. We will wrap up the rest of lesson number 6 next week. I haven't formalized the schedule for sure. My guess is going to be that we'll finish lesson number 6 on Tuesday, because I think Monday we're going to do the last lab that we have before the midterm. So tentatively plan to meet me back here Tuesday next week to wrap up lesson number 6. And you can always email me with any questions that you have between now and then. Before we close out the recording today, any last minute questions that I can address? So Christian asked about the due date. Lesson number 6 is not due this week. It's going to be due next Friday. The assignment might have the wrong due date written on it because I type those due dates in manually. I tried to correct them all, but if you know of places where it says they're due on the 28th, let me know, and I'll go in and correct those. But yeah, they're not due this Sunday. It's only lesson number 5 stuff that's due this Sunday. So lesson number 6 due next Friday in just over a week. Lesson number 5 and protein folding and disease, that's what's due on the 28th. Yes, you're welcome, Krissa. Happy to clarify that, and I will try to get that information out again. Yes, thank you for the reminder, Tien. Here we go. Here comes today's penguin. I have to give you a penguin after my computer crashed like 3 times, right? I'm just saying, for all of my friends who don't come to class or don't watch the recordings, whenever I do penguins anywhere, they're probably like, what in the world? This teacher is so weird. Yes, I'll give him some sunshine. We all need some more sunshine, don't we? Yeah, and the question about the midterm reviews, they are due next week as well. So they're due, I think what is it, the 5th? That's Sunday, or 7th, whatever day the midterm exam is due. So I think it's the 7th. We need to do those midterm reviews by the 7th as well. So we're giving you plenty of time to use them to study for the midterm exam. Yeah, and coming soon, next week we will definitely do a midterm review. We'll definitely do a histology review as well, because I know we all love those tissues, right? All right, well, I'm going to close out the recording. Please feel free to email with any questions that you have. I'm going to stick around in the classroom for just a bit, but this is a goodbye to our friends on the recording.